explain the molecular methods that are involved in MGOT?
The seeds need to be sown, and generally fresh leaves are used for DNA extraction, fresh juvenile leaves, because they give a good yield of DNA which is used in these reactions. There are protocols where you can even extract the DNA from the seeds itself. That again saves your 4 or 5 days, but the protocol is tricky and it is totally crop dependent. So first of all, you need a good quality of DNA, and then the DNA is used alongside of some other chemical components like DNTPs, which adds the extra nucleotides to the reaction, and a very critically an enzyme. It's called a polymerase, which is a Taq polymerase, actually derived from a Thermus aquaticus bacteria, and primers. Because when you come to primers, it's very critical part in the molecular GOT, because there are different types of primers used in the molecular GOT. Mainly the primers used are SSR and ISSR primers, and among them SSR mostly. So what happens in this reaction is the primer goes and sits, binds to the template DNA, and the enzyme amplifies the DNA. So from one copy, you get millions of copies of the DNA. So from that, now these are, SSR stands for a simple sequence repeats. So they are they are located in most of the parts of genome, and they go bind at a very specific location. So on the gel, you can tell that when you run alongside a male, female and a hybrid, you will see there is one typical profile or a single band for a male parent, and there is another sized band for a female parent, and you can see the both bands present in the hybrid seed. That's what tells you that, okay, this particular hybrid is coming from these two parents. That's the way of telling if it is a perfect hybrid. This is the trait purity that's been done.